Okay, so here we look at the idea of how we regulate metabolism, and I refer you to page 1153 in OpenStax, the two tables. I said, know these. There's no reason. It's a straight memorization and matching. This should be a review, looking at some of the things that we talked about back in endocrine. So just right here's the important ones, right? For catabolic, which catabolism, we think of this as our famine time, right? We're basically having to go in and use our stores. So we be cortisol, which allows us to do gluconeogenesis. We have glucagon, which stimulates the breakdown of glycogen, right? So this, you make new glucose. This, you take apart what you've stored up when you were in your feast state. And adrenaline, epinephrine helps you with the process of gluconeogenesis as well. So again, maintaining that glucose level and using other forms for energy is going to be very important during our famine state. In your feast state, which is defined by a lot of anabolic processes, you're using the stuff that you took in and using the energy that you've gotten in the calories to build muscle, bone, and store energy as adipose. So growth hormone is very prominent, an anabolic hormone, right? Helping you stimulate these. Insulin-like growth factor works in a similar way. Insulin, again, back from chapter on endocrine, insulin helped to direct a lot of anabolic processes. It helped you to move glucose into the cells so they could be used to power a lot of synthetic reactions. Testosterone and estrogen are all anabolic, highly strongly anabolic hormones, right? That get things built and utilize energy that you have available to keep on to do this in your feast state. All right, so energy is transferred again because you can't create it or destroy it. So that is the proper verb for this. This transfer is accomplished by moving electrons around, and electrons commonly come from hydrogen atoms. So we're going to keep our eye on this. Clearly, carbohydrates, lipids, proteins have a lot of electrons in them and a lot of hydrogens in them. Right? So oxidation reactions, and again, I'm trying to color code these consistently. These are taking things apart, and basically you are losing electrons and hydrogens when you are oxidized, okay? And the molecule is becoming smaller, so this is also something we talk about in catabolism, that you take larger, more complicated molecules and you get smaller ones in the reaction and release energy. The electrons will be transferred to another molecule because every time you have an oxidation, it is followed by a reduction. We're going to see here in a second. And I remark this because matter is stuff that everything's made of cannot be created or destroyed, and electrons are matter, so they can't just disappear. So reduction would be the molecule gaining or receiving this electron, and remember, this electron is a package of energy. All right, these reduction oxidation reactions are controlled by enzymes, enzymes that you mentioned back in A&P1, enzymes we've talked a lot about now in A&P2. So enzymes basically make chemical reactions happen, and now we're just looking at specific types of chemical reactions we call redox reactions. And enzymes are often assisted by coenzymes. We didn't mention any of those in the digestive system. It just wasn't necessary. But two very prominent coenzymes, and note that coenzymes basically, again, another type of molecule that assists an enzyme. Coenzymes are often organic molecules, and specifically they're often vitamins. So nicotinamide adenine dinucleotide, or NAD, that's the acronym, is niacin. Right? There's a reason you have to have so much niacin in your diet. And flavin adenine dinucleotide, or FAD, is from riboflavin. So we have to consume a certain amount of the vitamins so we can use them in these chemical reactions. But remember, I said vitamins are not taken apart by the body. So they are basically just being used to move these hydrogens and electrons that we're going to talk about. All right, so here's our carbohydrate, the fate of carbohydrate and carbohydrate metabolism in a nutshell. All right, again, remember that here's our glucose, which is 6 carbons, right? Organic molecules have carbons and hydrogens, and carbohydrates have oxygen. In fact, they're named because they have 1 carbon for every water molecule. That's why it's a carbohydrate, right? Simple carbohydrates are the mono and disaccharides, and complex ones are called polysaccharides. We recommended that you took in a lot of the complex. The complex ones are processed or broken down to the mono and disaccharides and monosaccharides by amylases. And the disaccharides are broken down to monosaccharides for absorption by disaccharidases, right? Maltase, sucrase, lactase. They were absorbed by facilitated diffusion and active transport. And I mentioned that here. So they're now in your blood, and they're being moved, transported in the blood to cells. Cells want this, and insulin tells them to take it up if they don't already have a permanent glucose transporter in their membrane. All right, so that's the job of insulin there. And in the cells, they're going to be used for ATP synthesis in a series of biochemical reactions we call cellular respiration, a metabolic pathway. So here's kind of looking at the cartoon of this metabolic pathway, right? With glucose is going to be broken down to produce as much ATP as possible. We have truly these 4 steps to what we call cell respiration, and they're shown here. The first step, if glucose is involved, is called glycolysis, because you're breaking down glucose. And when glycolysis is done, and this is done in the cytoplasm of all the cells in your body, because they all have the proper enzymes, you can make ATP, but you don't make much. All right, so that's why we can't stop here. And glucose becomes pyruvates as it releases energy, as it's oxidized. Okay, pyruvates then move into your mitochondria, and specifically this space in the mitochondria we call the matrix. And inside the mitochondrial matrix, the pyruvate, through other sets of enzymes, is converted to this structure we call acetyl. And we add another vitamin, another B vitamin, coenzyme A, to it. Right? And this just helps move it around. All right? So pyruvate becomes oxidized again to acetyl. It releases a carbon dioxide here. So we get rid of some of our carbons. That's why a 3-carbon becomes a 2-carbon. The acetyl is then the perfect starting point for what we call the Krebs cycle or citric acid cycle. And this happens in the mitochondrial matrix as well. And the citric acid cycle is a series where we start with this oxaloacetic acid, and we come back to it. The first product is citric acid. That's where it gets its name, citric acid cycle. We release, excuse me, some of these carbons as carbon dioxide. We harvest some of the energy as ATP. We do a number of oxidations and reductions in this process, and that's really what these this kind of yellow golden arrow is showing you, the electrons being released and moved. All right, so about 2 ATP are made here, about 2 ATP are realized here, not enough to live. So what happens now is all these electrons and hydrogens that were being stripped off of these chemicals as they were being oxidized are given to those coenzymes. The NAD here, NAD here, NAD and FAD here. And so these, coenzymes are really electron shuttles. They just bring all this potential energy, that they're carrying into this other set of reactions we call, the electron transport chain. And this is a whole series of oxidative phosphorylations that are done, all right? And this is showing you the electron transport. So electrons come in with high energy. They came in from glucose, pyruvates, acetyls. And in the membrane of the, or what we call the cristae of the mitochondria, these electrons start getting stripped off, and hydrogens get stripped off. And from them, we can make ATP, with the final electron acceptor being oxygen. This is the role of oxygen. All the oxygen we're going to talk about in respiration, system, all that oxygen from the atmosphere that comes in, it's all destined for this. That's its point. Carried by hemoglobin to all these cells, so it can be there in the end to accept the low energy electrons and hydrogens. All right, so this goes through it step by step. Step 1, glycolysis. You begin with the glucose, you end with 2 pyruvates. Again, a little bit more detailed here because this is just showing glycolysis. There are 10 reactions in total, right, as 3 phases. You don't need to know the 10 enzymes. It's just showing you what's really happening in more detail. What do you need to know? That 2 ATP are used, so we have to invest them, showing here, right? And there's the 2 drawn on the phosphates there. We phosphorylate. And 4 are produced, but since 2 were used, you only recover 2 ATP because 4 minus 2 is 2. So 2 is what you can use. So there's our 2 ATP consumed. We do produce 4, but that means the net is 2. 2 NADs will be reduced. So we're looking at that here and here. So here's the ATP production. Sorry, this is the investment, right? Energy consuming. Here's the NAD being reduced to NADH, receiving electrons and high energy electrons. All right, and here, of course, are the ATPs made. Happens in the cytoplasm, does not require oxygen. Again, ends with 2 pyruvates. All right, a few things to note, right? The reactants, so I want to spell this out. This is the kind of stuff I want you guys to know. You start with glucose, you have to have 2 ATP, you got to have 2 NADs, so 2 of these molecules of this coenzyme, and of course you have to have ADP and inorganic phosphate to make ATP. Products, 2 pyruvates down here at the end, 4 ATP, 2 NADH, right, and 2 hydrogens. So ultimately 1 glucose breaks down to 2 pyruvates, donates energy to create 2 ATP and 2 NADHs, right? So potential energy, usable energy. This is a high energy molecule like ATP, but cannot be used by the cell. So we have to go on. All right. So now if oxygen's around, we will, right, be able to harvest more energy. So this is a nice cartoon that says, okay, so here's our, again, our shortcut look at the idea of, here's our energy consuming part of glycolysis. Here's the NADH, right, the oxidation reduction, right? And here's the energy releasing. So this is all still part of glycolysis because here we end with 2 pyruvates. But this says, look, if you have oxygen around, aerobic, you will go on through this whole pathway and you will produce a whole lot of ATP. If you do not have oxygen, the reactions actually come to a stop, all right? So if we want to go on to get more, we call this, we have to have oxygen. Without oxygen, these are the anaerobic pathways. And what it turns out that happens is pyruvate here, right? So here's the end of what we consider glycolysis, gets converted back to lactic acid or lactate. So pyruvic acid is actually then reduced. So we have to remove the electrons from NAD and give them back. All right, so we still got our ATP, right, that we formed here, but we can't go on and we can't do anything else right now with this lactic acid. And so this is a very effective pathway so that you can still get ATP right here when oxygen is not around. It's just, it can't be used for long. And this was mentioned to you guys back when you talked about the muscular system, right? And all the ways that muscle is such an ATP dependent tissue that there are times when you can't get enough oxygen to the muscle, right? And it uses up all its myoglobin. And so you're left with still needing ATP, so you don't just stop because you need ATP to contract and to relax. And so the answer to that problem is that you make lactic acid. And that lactic acid unfortunately stops, right, so that we don't harvest and get all the usable energy, but we at least can still get ATP. All right? Now, in the next video, we'll talk about, well, when we do have oxygen and what we're going to do in that circumstance.